All right, welcome back to The Explainer. Today, we're diving into the absolute cutting edge of structural biology. We're going to zoom all the way down to the atomic level. Why? Because we need to see exactly how AlphaFold 3's new AI architecture is literally reshaping our understanding of life itself. It is wild. Let's just kick things off with a totally mind-bending number, 70%. AlphaFold 3 hasn't just tweaked a few academic models here. It has actively reduced the time it takes to screen cancer target chemicals by 70%. Seriously, what used to take years of endless, grueling trial and error, it's now happening at lightning speed. It's a total game changer. Okay, let's break this down. First, we'll look at the molecular miracle. Then enter the Pairformer, followed by the diffusion dance. From there, we'll be decoding the black box. And finally, exploring the evolutionary anchor. Let's dive right in. Part 1, the molecular miracle, unlocking the dark proteome. So for decades, molecular biology was essentially just this observational science, right? You needed massive, super expensive equipment just to painstakingly look at what was already there. But that whole paradigm, it's shifted entirely. It is now a predictive, engineering-driven discipline. Just look at this incredible shift in data from the Sovereign Tech audit. Human proteome structural coverage, it jumped from 48% to 76%. And this next part is just wild to me. We went from having over 5,000 completely unsolved human proteins down to just 29, barely 24. Furthermore, the total mapped protein structures absolutely exploded from the strict limits of physical experiments to over 200 million via the AlphaFold database. It's basically like searching for a needle in a haystack for decades, and suddenly someone just hands you a giant magnet. And because of this AI-driven orchestration, the so-called dark proteome, that mysterious residual chunk of human proteins that structural biologists simply could not map, that has completely collapsed down to just 10%. We are essentially flipping on the lights in the darkest corners of human biology. Part 2. Enter the Pairformer, upgrading the architecture. So how exactly did they do it? Well, to achieve these monumental numbers, the underlying AI system had to fundamentally evolve. AlphaFold 2 relied on the Evoformer architecture. And hey, that was a massive breakthrough in its own right, for sure. But it was mostly designed to solve the single-chain protein folding problem. AlphaFold 3 steps the whole thing up. It completely replaces that earlier model with the Pairformer, which is this brand new architecture specifically built to model complex, multimolecular assemblies. We aren't just looking at one protein in isolation anymore. We're looking at how vastly different molecules actually interact and behave together in real time. Think of it like a highly complex 3D molecular dance. Before the AI even attempts to figure out where the individual atoms actually go, the Pairformer directly models the interactions between the amino acids. It maps out the tension, the proximity, and the relational geometry between them. It's essentially establishing all the rules of the dance floor before the dancers even arrive. Part 3, the diffusion dance, computing atomic coordinates. Now this is where the magic really happens. Step 1, the Pairformer models those complex pairwise interactions we were just talking about. Step 2, the AI applies a brand new diffusion-based generative model. Imagine starting with this cloud of completely random molecular static, and the AI just iteratively clears the fog away, orchestrating and refining the shape at a microscopic level. Finally, in step 3, it computes the exact 3D atomic coordinates. It's literally a breathtaking computational ballet that shapes proteins, DNA, RNA, and ligands with near-experimental precision. And because this rapid atomic computation only takes minutes instead of months, it has completely democratized structure-based drug discovery. Right now, over 500,000 research projects are being supported globally via the open-access AlphaFold database. This level of access means researchers in developing nations have the exact same predictive power at their fingertips as the world's absolute most elite pharmaceutical labs. So for you and me, what's the takeaway? The timeline for discovering life-saving medicines just got radically shorter no matter where on Earth the research is actually happening. Part 4, decoding the black box, mechanistic interpretability. But here is the million-dollar question, right? How does AlphaFold 3 actually know how to fold these proteins? Is it just, hallucinating structures based on statistical noise? If we're going to trust this AI with our lives and our medicines, we absolutely have to know exactly how it arrives at these atomic coordinates. So to break open the black box, researchers treat the AI almost like a physical system, using what's called mechanistic interpretability. Using a really cool technique called causal activation patching, they can isolate specific pathways inside the Pairformer. It's basically like tracing the AI's internal wiring to see exactly how it makes a single decision. And this allows researchers to audit its structural priors, which are essentially its foundational assumptions about how biology actually works. We are quite literally watching the AI think, step by step, just to ensure its logic is firmly grounded in physical reality. And the audits revealed something absolutely amazing. As the AlphaInterp research explicitly notes, the model behaves similarly to an advanced fold recognition system, meaning it isn't just randomly guessing shapes hoping for the best. It acts as an incredibly advanced engine, strictly following the folding logic that is embedded deep within the universe itself. Part 5. The evolutionary anchor, phylogenetic diversity. So where exactly did it learn those deep structural rules? Well, the audits revealed that the AI's pinpoint accuracy relies heavily on phylogenetic diversity, not just the depth of data alignment. In simpler terms, it relies on the vast, ancient tree of life. The AI actually compares how a protein folds in a human versus a fungus versus some deep-sea bacterium. It is the ancient, comparative evolutionary history of these massive molecules that anchors the AI's entire structural logic. Which leaves us with a truly profound thought to wrap up on. What does it mean for the future of structural biology when our most cutting-edge artificial intelligence relies entirely on the deep, ancient evolutionary history of life? To engineer the absolute future of human medicine, our AI literally had to learn from billions of years of biological trial and error. Thanks for exploring this with me, and I'll catch you in the next explainer.